Well, we're really excited to get this session started. I know it is after lunch, and we're going to do our best to captivate your imagination and talk about the future of brain-computer interfaces today. We got a really exciting setup with some fantastic speakers. I think, we've all been excited about brain-computer interfaces. It's kind of like the holy grail in some sense for many of us that grew up interested in neuromodulation and in the brain itself. So, and there's a lot of media right now. Some people are good about getting media attention, and so we're excited about where things are heading. But it's a good time to talk about brain-computer interfaces. I think one thing that Ian and I, and he'll introduce himself in a second, and maybe I should introduce myself. I'm David Darrow. I'm a neurosurgeon scientist here at the University of Minnesota. Have a lab here and a lab at the county hospital where we focus on spinal cord injury and neuromodulation with a lot of partners like Dr. Nedoff and others in the crowd. And brain-computer interfaces are really exciting, but I think one thing about brain-computer interfaces that's not obvious to everybody is that they come in all different shapes and sizes and with different goals and purposes. So I think we're going to see a little bit of that today with different approaches that people have taken to try to bridge a translational gap, but with, in the context of this larger idea of tapping directly into the brain and doing what the spinal cord already does pretty well.
Perfect. I'm mic'd up.
Okay.
Good. Hi, everyone. My name's Ian Burkhart. I'm the founder of the BCI Pioneers Coalition, Brain Computer Interface Pioneers Coalition, since we all love acronyms. I myself had a brain computer interface and was implanted for a trial from April of 2014 through August of 2021. We're using that to restore hand function. I have a C5 Asia A injury from about 14 years ago now. So about 4 years after my injury, I was looking for more. I kind of hit a threshold of routine inpatient and outpatient therapy, and I wasn't happy with where I was in my life. So I joined a trial between The Ohio State University and Batelle. You'll hear a little bit more about that later by one of the other panelists, but I wanted to talk a little bit about kind of what really is a brain-computer interface, because a lot of people use the term, I think, a little vaguely because it is such a buzzword, which is great. The way that we at the BCI Pioneers Coalition define it is essentially using it as a pipe from the brain to some sort of outside device. So whether you're controlling a computer, controlling a robotic exoskeleton, or something that then is giving feedback directly to the body. What we generally say isn't a brain-computer interface is something that's operating independently and just like DBS doing stimulation of the brain. Now, closed-loop DBS is a little bit closer because it is recording information from the brain and then passing that right back to the brain to make a change. There's also different types of brain-computer interfaces, whether you can do stimulation or just recording of the brain. To date, most of the studies have been all done in the motor cortex, some in the sensory cortex, but it's all about motor control and then communication. And so that's going to be really the first indication that I think will get approved for the public to use. Now, you can also use it for controlling muscle stimulation like I was able to use. For that, it was an external muscle stimulator on the forearm. You can certainly use it to control a spinal cord stimulator, whether that be implanted or transcutaneous, robotic devices or exoskeleton. But I definitely see that communication is probably going to be the first one that's approved and actually gets reimbursement for. But then again, I don't have a crystal ball, unfortunately. But I do think it's really important to highlight also that there's all these different types of both invasive and non-invasive, so wearable or implanted devices, and you need both for patient choice, if someone is willing to have surgery or not, and also for the different types of applications that you want to use it for. If you really need high resolution input and data, or you need to be able to stimulate the brain, that's most likely going to be something that is invasive. If you need something that's more of a switch type control, you can get away with something that is external and wearable, and that helps patients because it lets them choose something that works better for them and their lifestyle. There's also a lot of talk about kind of ethics and public perception of brain-computer interfaces. A lot of people think, and I think it was on Tim's slide earlier, of, Neo from the Matrix and the $6 Million Man. And, we do see a lot of that, and people think, oh, you're putting a chip in someone's brain, they're going to be able to take over the world, or they're going to get controlled by someone else. That's not where we're at right now. For better or worse, it's still pretty infant stages of what we're able to do. But we're able to use these devices to really help people with disabilities retain a lot of quality of life. And I think that that's something that's really important. Going back into the ethics and that side of things, there's a lot of ethics involved around data privacy and who owns that data, how much of the data is really necessary to do the intended task. And then also, as a field, we need to make sure that these devices have longevity in mind when they're being developed. It was mentioned once again by Tim earlier this morning. You don't want to have a device that in 10 years you can't use because you can't get hardware or replacement pieces, especially if you're talking about something that's implanted in someone's body. You need it to be able to last for their lifetime, or at least be upgradable and replaceable to the extent that you're not subjecting people to further surgeries. But I'm really excited about the panel today. We have 3 kind of experts in the field. I think Dr. Darrow is going to introduce our first one now, and we'll go from there and then have questions at the end.
We are very excited to have Dr. Banerjee come, sort of homecoming back to the University of Minnesota. She is already well known, but she's the CTO at Synchron. She's been all over the media and did her PhD and master's here in electrical engineering. Synchron, for those of you that don't know, is the first company with FDA IDE approval and is currently conducting their clinical trial for implantable BCI technology. Dr. Banerjee has a significant experience in many conditions such as Parkinson's disease, chronic pain, and urinary incontinence. She is passionate about bringing together people with deep expertise and creating team culture that drives impactful neurotech innovation and makes a difference in people's lives. She's going to be talking today about developing a scalable, minimally invasive brain-computer interface platform for today and tomorrow. Hopefully we can all welcome Dr. Banerjee today.
Thank you. Well, it is a pleasure and an honor to be here. Very fun. Yes, I am from Minnesota, and I had to put on my Gopher pride. So yeah, I lived here for 20 years, and for those of you that don't know my story, I was at Medtronic Neuromodulation, and then had this opportunity to go to a startup, and Tom Oxley wanted to set up in New York City. And I think I was like, I don't know, I've always wanted to live in New York. I don't know what it'll be like to hire in New York. They have a couple years of funding, and it's like still COVID, so my husband was working remote, and we were like, I don't know, I think we could do this. So we kind of just went and took a chance, and it's been quite the ride and interesting experience. But I've just returned to live more in Minnesota. I'm still obviously at Synchron, able to work remotely, but just for family reasons. So anyway, so Minnesota is home and my roots as well. So of course I'm an employee and stockholder of Synchron, and I'll be talking about devices that are investigational. So this has been a really nice conference talking about, this isn't just about the technology, but translationally, like what are we trying to do that creates value? How do we understand our clinical trials? How do we understand compelling endpoints? How do we understand reimbursement and ultimately, value for our customers and our patients? So that's kind of the direction that I'll be talking about. So what is the unmet clinical need with BCI? So we are focused on people with severe paralysis. And people will ask me, are you only focused on ALS? Are you only focused on spinal cord injury? And ultimately, we're trying to restore capability for people to communicate when they have severe paralysis. And that paralysis can come from many different things. So spinal cord injury, as I mentioned, multiple sclerosis, some of these are going to be neurodegenerative, meaning that they're progressing over time, but some of them will have kind of an acute onset and have stability. So there's over 5 million people living in the US with paralysis. So we have Ian here, sitting here today, and I actually have him in I thought, that's not you. Oh, sorry about that. Yeah, I thought that was you because it's a kind of a blurry picture, but yeah, that's another gentleman who was an early pioneer, but I love the BCI Pioneers initiative that you have, and it is true that these are pioneers that are trying out the research devices. And so we have kind of 20 years of history here from a 2006 paper of the control of the robotic upper limb using the 96-channel Utah array. And then showing some of the more modern research coming out in 2023. And you can, there's amazing things coming out in terms of state-of-the-art text generation, 62 words per minute. But one of the challenges here, and I was just talking to David McMullen at the GRC Neuroelectronic Conference, and he was like, one of the things in BCI is for every one of these publications, there's 40 scientists behind the curtain that are kind of making this happen. And so that's kind of been one of the barriers of translating this highly complex technology. Some of these are not fully implantable. They are high bandwidth recordings that are taken through a pedestal that's penetrating the skin and then connecting up to like a rack of equipment. That's not so scalable for people in their daily lives. And then troubleshooting. So in terms of stability and being able to use this, you need highly skilled researchers actively involved, and it's too complicated for caregivers to support their loved ones with this technology, and even to deploy to like occupational therapists to work with people in this domain. And then stability of the signals, so do you have to do ongoing calibration? And so kind of recognizing that for BCI to provide meaningful benefit and to be scalable, it must enable user autonomy and continuous functionality. It can't just be done in a laboratory with a scientist present to help that person. And so Nick Ramsey kind of put together these 5 tenets, and that's kind of what we're building upon. And so many of you know Dr. Ramsey and his lab, and I think Tim Dennison presented the work that was done there with the first fully implantable BCI system in 2016. And these are kind of 5 key tenets to boil down. So easy to learn, low cognitive effort, ease of use, low recalibration, immediate use, that it works in the first session, and always available and stable use. I also will reference this kind of foundational seminal work. As Tim mentioned, she's allowed the usage of her name, Hanukkah, and she's been using this system for over 7 years. And she can use this system with minimal assistance, and she really liked it because she could use it outside. And so when you compare it to things like eye tracking and other assistive devices, she found benefit over this. So she talked about her eyes being too slow for her eye tracking system, and without the BCI, she would be at a loss for words. So what's unique about Synchron? Synchron has the endovascular approach. So the founding of Synchron is by Tom Oxley, Nick Opie, and Rahul Sharma. And they come from the endovascular space. Rahul Sharma works in the cardiovascular domain, and in that domain, there is a lot of innovation in deploying devices through the cardiovascular vasculature. And over the last 10 years, there have been a lot of stent-like devices that have been deployed in the brain for treatment of things like aneurysms and strokes. And so the Stentrode was developed to be a stent-like device, but that provides that electrical connection or that neural sensing functionality. So it's a stent-like device that seats at the top of the brain and then is connected to a lead. The lead comes out of the blood vessel and then is tunneled subcutaneously to a device that's in the chest. All right. And then one of the key elements is stability, that what I mentioned earlier. And because it's an endovascular approach, one of the nice things is that the stent actually endothelializes into the blood vessel. This is a common thing that happens for stents, and the endothelialization actually provides a very nice stable environment for the electrodes to sit in. Also, then you kind of avoid some of the tissue reaction elements that happen when you're in direct contact with neural tissue. The deployment of the device is in the superior sagittal sinus. The superior sagittal sinus is that vessel that's right at the top of the brain, and that happens to be a great location for where your motor cortex is. So what this is showing is imaging showing where those electrodes are with respect to key areas of function where we want to do sensing, such as the primary sensory cortex and the primary motor cortex. So how do we think of clinical meaningfulness? So in able-bodied people, you think about your motor intention. It goes from your brain, the primary motor cortex, and through your brainstem to the spinal cord, and then to your peripheral nerves and your muscles, and eventually you touch your phone or your computing, and you interface with devices and computers. There are a lot of different conditions that can cause paralysis, as I mentioned, and that can be because of dysfunction anywhere along this pathway. So what this image is showing is that when you have a stroke, for example, that might affect your brainstem, and therefore that's where the signal pathway is terminated or incomplete. Spinal cord injury might be further down. The peripheral nerves, that might be a different condition, and then eventually maybe there's a muscular disorder that's causing the issue. And then this ecosystem that we've created that we use for communication, for interacting with the world, is now not available to people that have these conditions. And if you think about it, it's not just texting or communication. Nowadays, with the Internet of Things, we can turn off the lights. We do so many different things. We can interact with our environment if we have that capability to interact with devices. So the value of brain-computer interface is restoring the transmission of the motor intention. And so you're thinking about, and you have that motor intention, and right from there, the Stentrode is extrapolating that information and creating a digital motor output. So instead of physically connecting with a device, you're now kind of thinking about it, and digitally, that's what's appearing on the device. And that's how you get the control signals. One key benefit of that is, just in terms of what data we're collecting, those are physical things that you would be seeing anyways. You would be seeing me interacting with a device, right? So those are the types of things that we're collecting. And if you couple these digital motor outputs with instrumental activities of daily living, then you can create a metric or a measure of how someone can function independently within society. The instrumental activities of daily living are coming from the domain of occupational therapy, and so these types of things, communication, can you manage your own health? Can you interact with your environment? Are you able to do your own shopping, manage your own finances, entertainment? These are all the ways and the scales that are used in the domain of occupational therapy. All right. And then another thing I'll introduce is our system is coupling with a pretty vast ecosystem of accessibility technology. So this video that you're seeing there, where it's highlighting different boxes, it's highlighting different groupings that makes the user interaction and user interface more simple, is something that's on our devices today. If you look on your phone and you look for accessibility modes, you'll be able to use and create these things, and they're used for people with varying needs. So if you couple this with the BCI, you can see that you're making the user interface a little more simple for people to interact with. This is one of our patients, our command patient 2. It's given us permission to share this video, and it shows how we're using that type of user interaction for him to be able to convey his pain score. So he's working with an iPhone. You can see he's very severely paralyzed, and this is an iPhone that's sitting in front of him, and he's using his BCI to interface and communicate his pain score. And you can see a little smile on his face. It's one of the residual aspects of mobility that he still has that is a nice communication for us to see. This is one of our patients in Australia, and this is a different user interaction model that he's using to manage his finances. So you can see like our device has, sometimes we talk about lots and lots of electrodes, lots and lots of capability, rack mounts of equipment. We're kind of going with a model that's simplified, and let's understand the ecosystem that's out there in terms of user interface and interactive models that can create a compelling user experience. This is the same gentleman, and he's using a system with large language models. So he's using some amount of predictive texting to be able to improve his communication. And you can see a smile from him as well. Oops. Sorry about that. And this is one of our US patients playing Pong, and we're able to, he's able to control with a fair amount of continuous control the paddle for Pong so that he can play. Which of course is one of the seminal computer scientist games that has some good geek street cred, I think. All right, so what is our journey of clinical translation? Where are we at? So the journey of Synchron starts around 2012, and I was at the GRC Neuroelectronic Conference. They said on average in this space it takes about 17 years to get to the journey of pre-market approval. And for Synchron, it's going to be about that. Starting back in 2012 with preclinical feasibility work, the first study that was published in Nature was in 2016, showing that there was sensing that you could sense from a blood vessel in a large animal model. After the publication of that paper, then the founding team, I like to call them the founding team in Melbourne, Australia, then looked at how to create a system that was amenable for implants in people. And then in 2019 and 2020, 4 patients were implanted in Australia, and that was the switch trial and the first in human that was completed in 2021 and has been fully published. I started with the company about 2.5 years ago now, and it was kind of at that time that they were having success with the switch trial. They got FDA IDE approval to start the command trial in the US and build up a team in the US. So we're now a very cross-disciplinary team of 80 people. So we've had a significant amount of growth, and then we're working on the command trial with the six people that are in the US. And I have this kind of colorful diagram on the right side. And I enjoyed Tim's presentation where he had that nice circle diagram. And he talked about the ecosystem that you have to think about as you kind of grow. And you grow from a technology space where does the technology work? And the baseline questions you're asking is does the technology work? Is it safe? And I kind of also like to say that it's difficult to get your voice of customer until someone's implanted. So you kind of always are thinking about the patient and putting the patient in the center, but until someone's implanted, that's like a really rich time of learning. And so it's kind of with those first 4 patients and then those 6 patients that we're in a really rich time of learning and understanding, and that enables us to create something more robust and more compelling. And so, we're in that time, why do you grow from like, a 10-person team in university research to kind of an 80-person cross-disciplinary team is now like as the time where we have to really consider to grow, to create value, to create a product successfully. We have to be thinking about regulatory, about reimbursement, about clinicians, about the surgical aspects of the procedure, about the patients, the caregivers, all of those aspects of it. And all of that kind of has to come together to create a compelling product. And so that is kind of the journey of growth that we're on and kind of where we're at in our journey. I think that's all I've got.
Wow, that was amazing. We're going to have the panel at the end, but you don't have to sit up with us and hurt your neck looking at the screen like we are. All right, well, that was an amazing start. Next, we have Dr. Galvez. Dr. Galvez is a postdoc neural engineer specializing in brain, spine, and brain-machine interfaces for transformative neurorehabilitation therapies. She comes to us from Lausanne, from, I guess, the NeuroRestore lab or group from EPFL in Lausanne, and has been really interested and has expertise in decoding brain signals, spinal cord stimulation, and developing neurotechnology to empower individuals with spinal cord injuries and stroke survivors in regaining motor function. We're very excited that she was able to come all the way across the pond to talk to us today about brain-controlled spinal cord stimulation to restore walking after spinal cord injury.
Thank you very much for the introduction. So good afternoon, everyone. I'm Andrea Galvez. I am a postdoctoral researcher in NeuroRestore. And it's a big honor for me to be here representing a big group of scientists, engineers, and students of NeuroRestore. And in this session of brain-computer interface, I will be presenting the brain-controlled spinal cord stimulation to restore walking after spinal cord injury. So let me introduce you a bit who we are. We are a center of neurorehabilitation funded by Professor Bloch and Professor Courtine in Switzerland, in beautiful Lac Léman. And we have 3 platforms. The first one is in Geneva. It's our preclinical platform. The second one is a translational research platform in Freiburg. And the third one is the clinical site at the hospital at Lausanne, where actually I am. So let's dive more into science. And as we have seen and we all know, when we think about walking, the brain sends descending commands from the motor cortex to the different regions of the spinal cord. And after a spinal cord lesion, this communication is interrupted, which leads to paralysis of the muscles below the level of the injury, having, of course, devastating consequences. But what we know is that the neuronal circuitry below the level of the injury is still intact, of what we call in a dormant state. So one of the strategies to alleviate paralysis has been to stimulate electrically the spinal cord below the level of the injury. More specifically, the circuit is responsible of activating the leg muscles located in the lumbosacral region of the spinal cord. And of course, there has been a lot of pioneer work done in rodents around the world. And for example, here we could see this rat model of paralysis, and the animal receiving spinal cord stimulation continuously, then the animal regains this capacity of walking and stepping. And once the stimulation is off, well, the animal is not able to step anymore, and it's paralyzed. So very briefly, what are these mechanisms of spinal cord stimulation? So here I will present you, this is a cross-section of the spinal cord. And we could see the different afferent fibers in red coming from the skin and the muscles. And what we could see is that these connections can enter through the dorsal root entry zone, connecting directly to the motor neurons or indirectly to a chain of interneurons. And preclinical studies and in silico studies have shown and revealed the underlying mechanisms of ES. And what we know is that when we apply ES, when we apply ES, we are able actually to activate the motor pathway in an indirect way. And the motor pathway is actually indirectly activated by the recruitment of these proprioceptive fibers, that is the large diameter fibers, which are called the A-alpha fibers. So how does this apply to humans? As you know, walking involves a very complex sequencing of activation of muscles in our legs, the different extensors and flexor muscles. And we have explored how normal locomotion elicits the temporal activation of these specific motor pools, depending on the gait cycle. And you could see here in this graph, in the x-axis, we're showing one gait cycle. And in the y-axis, you could see the different levels of the spinal cord. And you see these different hotspots of activation that are actually activating depending on the gait cycle. And we can see and identify then the circuitry that actually is responsible of locomotion and allow us then to understand or to see that these specific hotspots, we are able then activate them with stimulation and probably restore locomotion. And this is our aim, to be able to mimic this natural activation of the spinal cord, targeting these specific hotspots during walking. And this is what we call the biomimetic spinal cord stimulation. So how was this path to humans? So I have very briefly shown you that we have tried to understand this different activation of the hotspots in the spinal cord, and that we want to target these hotspots at the lumbosacral region for walking, that we are limited in technology and that we are going to use at the time the available technology, but we needed to actually enable this technology in very precise timing with this Swiss precision. And here it comes the role of us as engineers. So in the first patients, we needed to adapt the technology from Medtronic. So this is the 565 paddle lead connected to a stimulator. And then we needed to communicate, and in order to communicate with this stimulator, we use an antenna and a patient programmer that communicates through electromagnetic induction. And thus this patient programmer received the stimulation commands coming from an external computer, and we have built this software that is able, let's say, to allow us to design these different stimulation programs to be sent wireless via Bluetooth to the stimulator. So the neurosurgeon implants a lead of 16 electrodes in the dorsal part of the lumbosacral region of the spinal cord. And this lead is then connected to the stimulator. And then our role then is to be able to design the different, and to be able to target these different hotspots to design the different stimulation programs. So for instance, example of a configuration of a hip flexion in a specific, let's say, waveforms and frequencies, and this is one example, or then to target full leg flexion, or weight acceptance during the gait cycle, or propulsion. And I'm just showing you very quickly some examples of the different, let's say, parameters we modify for these building blocks of stimulation. And of course, it's an example, but this is done for each patient, so it's built during a phase of mapping. So here's one example of one of our participants with spinal cord injury. In the left panel, you could see the motor scores, which are 0. And this patient is attempting to step with a bodyweight support system. And you could see lower in the panel the different EMG activity of the muscles. And you see that there's no activation due to the lesion. And there is no, let's say, response even with the attempts. And then here, I could show you the effect of the biomimetic spinal cord stimulation, and you could see the participant trying to step, and you could see the different activation of the muscles, the burst of activity, the effect of the stimulation. But when we turn off the stimulation, the participant loses this capability. So in order to be able to arrive to that point of these building blocks of stimulation, we needed to do these very long-lasting sessions, very time-consuming, when we needed to test all the possible, let's say, parameters of the cathode, the anode, the different waveforms, pulses, optimizes per muscle group. And this was a process that was actually taking us weeks. And, well, it was not the most comfortable, of course, for the patient, and it required all of us, many engineers, to be involved to be able to design these configurations. So after these years, what have we learned from these first steps in humans? So the first thing that we need to highlight is that even though we adapted that technology, not designed for it, we knew that we needed a purpose-built technology. So we needed a technology that allowed us to unlock all of these world of possibilities to be used as a neurorehabilitation therapy. And of course, here it comes our key partnership with Onward Medicals. It's a technology developed, purpose-built, the RCM platform in collaboration, of course, with NeuroRestore to connect, and then we'll see to connect the BCI technology. Another thing that we have worked during these years is to develop an intraoperative pipeline which guides the neurosurgeon. And first we do this high-resolution MRI, then we merge with the CT scan of the spinal cord, and we also do a functional MRI where we stimulate the different tendons using some vibrators. And with this, we merge all of these, let's say, information to be able to guide the neurosurgeon to have the best position of the lead that will trigger the different hotspots in the spinal cord. We have worked and implemented on an AI-based configuration algorithm that allows us to test all of these different configuration settings, as we have 16 electrodes, 9 frequency, 10 amplitudes, 2 different pulses. This gives, very briefly, more than 6,500 combinations that we were not able to explore possibilities. But today, with this new approach, with this new technology, we are able actually to, within a very few minutes, and within the same day, we're able to get these configurations. So for instance, a knee extension, we will have the optimized configuration in about 5.5 minutes, a leg flexion, 2 minutes 40, and an ankle extension, 6 minutes and 10 seconds. Just to tell you briefly that what it used to take us weeks, now we are able to do it within a session. And, well, it has really opened a new door, let's say, of possibilities. So once we have these configurations, we use these building blocks of stimulation, and we combine them, and we put it in this software that we have used. So these are like the stimulation blocks, or what we call the stepping modules, that are the programs that the patients will use. So in summary, we have implanted 9 participants, 6 of them with incomplete spinal cord injury, and 3 of them with complete injury. And after 5 months of intense neurorehabilitation, we observed in vast majority of the participants neurological recovery. In the best cases, we observed recovery independent walking in parallel bars, and the least affected participants recover the most. And the participants with more severe lesions are those who recover the less. So I have shown you this technology based on these building blocks of stimulation, and we have actually pushed this technology to adapt it to a way that the participant is able to use it and to trigger it himself. So here, this is a video of this participant that is able to click some buttons in the walker, as you see, and he's able, every time that he clicks the button, to trigger the stimulation of the right or the left leg. And of course, this gives a sense of control for the participant, but yet it's an artificially triggered, let's say, technology. So then we question if there was a way that we could trigger the stimulation in a more natural way. So here it comes, our project connecting the brain to the spinal cord again. So our idea to improve this control over the stimulation is the use of the brain-spine interface, and we want to record from the motor cortex these intentions of stepping, and then link it to specific stimulation programs. And it's not a new idea. We have shown it in the past. The lab has published in a non-human primate this model of spinal cord injury when we record from intracortical electrodes over the motor cortex, the spiking data, and we use a decoder algorithm to interpret these signals and to send them back to a stimulator to be able then to stimulate in the different gait phases. And very quickly, an example, this animal has a hemiparalysis, and you could see the animal without stimulation. He's not able to perform stepping. When the BSI is on, you see him regaining this capacity of stepping. Again, the stimulation is off, and he loses, let's say, this capability of stepping. So we have shown that this concept, this proof of concept, was possible in non-human primates. So we wanted to make this digital bridge to bypass the lesion and to allow the patients to reconnect their thoughts with their legs again, and to have, more importantly, a gradual, voluntary control of walking. So here comes also our collaboration with Clinatec, a team in Grenoble in France, who have been developing for more than 10 years these brain implants, and these brain implants have very specific characteristics. So these implants have a diameter of 5 centimeters. They have been also implanted in 3 participants that have been using them for BCI to control an exoskeleton. We have chosen this technology as we have found it a good compromise in between minimally invasiveness and the surgery for a neurosurgeon is actually the replacement of the bone by the device. This gives the participant also an opportunity not to have any external connector, as it is what we call a passive implant. Now, the price of the passive implant is that the participant needs to use this helmet, what we call the antennas, to be able to communicate to the external world that I'll show you in another video. So I will introduce you our first BSI participant, his guardian Oscar. He came 6 years ago to the lab, and he joined the first clinical trial. He has a C5, C6 incomplete lesion. And this is a video in 2017, and he arrives to our lab. So the first thing that I want to highlight is that he's using a body weight support system, and here he's attempting to walk. We'll just reproduce again the video, and you could see that he doesn't have what we really call stepping capabilities, and neither this moving forward possibility. So he joins the clinical trial STIMO. He undergoes the 5 months of neurorehabilitation. He arrives to a plateau, and he continues in this extension period. It means that he's able to use the technology at home for rehabilitation. But then he is invited in 2021 to the BSI clinical trial. So in July 28, he's implanted with the brain implant. And here I'm showing you the post-CT scan image, and we could see here, and we could highlight the coverage of the motor and the sensory cortex. So these 2 implants are on top of these regions, so we are covering the motor and the sensory cortex. As early as the 2nd day, we started to test the system. We trained a decoder when he needed to follow some cues shown by an avatar. And then the bar shown just next to the avatar, you could see the feedback. So when the avatar here is showing hip flexion, he's following. You could see the performance of the decoder showing the feedback in the red bar. So in very few minutes, he's able actually to, we are able to build a very good decoder that we are also being able to use it when he's standing, for instance, here in hip flexion, and also for walking. So I will go here to show you. So here I'm representing in this grid of in the brain the different features that we use for the decoding, and then we see in the triangle the different features and the actually decoding outcome. So here the participant, the task is that he's going to do a hip flexion. So we will see how he will attempt. You see the decoding features going to the right, and then down in the panel you see the triggering of stimulation. Again, he goes to the idle, he will attempt, you see the features and triggering the stimulation. So we are able to translate these motor intentions into stimulation commands. And what is importantly is that he has this voluntary control of this modulation. It means that we have set up before the minimum and the maximum amplitude of stimulation, and with his intention, we are able with the decoder to decode the state, meaning, for instance, the hip flexion, and he's able to modulate that intention of the stimulation within pre-set up parameters. And here in this video, what I'll show is he's walking, and I represent the brain signals in the spectrogram, this graph just below. And I'm showing just what you see, these flashes, red and black, that is in single trial, you're able to identify these brain signals. And these brain signals, single trial, are then interpreted by the decoder. And the second panel, I'm showing a graph of the outcome probability of the decoder of the different states. And this probability curve is then translated of what we call stimulation commands. And here it's also where I will just grab your attention that it was one of the game changers that we changed the way we were stimulating. We are translating the probability curve of decoding into stimulation commands that updates every 200 milliseconds. And this is what we gives the more natural and smoother way of stimulation of the muscles. And this is what we say, that the brain-spine interface allows the participant to have a more natural walking. As you could see, the movements are much more smoother due to the way of this translation of the signals of the brain to the stimulation. But not only, and most importantly, is not the smoothness of this natural, let's say, characteristic of the way he's walking, but actually the capability the brain-spine interface gives to the participant to adapt to the environment. And this is also a game changer, because this allows to have a technology that is able to go outside of the lab. It's a participant that could use a technology to be able to be in real-life settings, to be able to overcome obstacles as he voluntarily can have a maximum, minimum stimulation to have a step, and to navigate in different directions and with the system. So this brain-spine interface we tested, of course, in indoor and in outdoor. And one of the main points that the participant was expressing was this way of feeling more free and having this reconnection with his legs. And we also had the participant got neurological recovery at the end of the clinical trial. As I've mentioned, this participant was first in the first clinical trial. He gained a neurological recovery, and after 2 years, he arrived to a plateau of recovery. In the third year, he joins the new clinical trial, the BSI clinical trial, and at the end of the rehabilitation, we do again these clinical measures without the system, and he has this neurological recovery in the motor scores. We were able also to embed all of this system in a walker, in a laptop, which allows him to use it independently back home. So he used the system in the rehab center, and now we are doing trying to push, let's say, this system to do his daily life activities. More importantly, and one of the things that have been mentioned during the day is that the same model has been used for more than 6 months. I check on him and connect in monthly basis just to check up on the system, and we are also able to do online calibrations and to build new decoder in distances. He's in the Netherlands and I'm in Switzerland. So our next steps was very exciting, the new generation of the technology with Onward, which will allow us to embed many more clinical trials, and our perspective together with Onward to develop the next generation of this universal plug-in technology to bring together all of these developments. And with this, I want to thank you for your attention. Thank you for the invitation.
Yeah, thank you so much. Super exciting, Dr. Galvez. Thank you for telling us about all the cool things that you guys are working on over there in Switzerland.
No, Next we have Dr. Dave Friedenberg, who is a principal in neurotechnology and the team lead for the machine learning and AI team in the health analytics group at Batelle. He was actually one of the individuals who was instrumental in working on my project using a BCI to control muscle stimulation that allowed me to become the first person to restore hand movement after a spinal cord injury. He's also going to talk a little bit today about using closed-loop functional electrical stimulation to drive neuroplasticity after a spinal cord injury in a non-invasive manner with a new project that he's been working on. So as soon as we get these slides up here, I am more than happy to welcome Dr. Dave Friedenberg up to the stage, and we'll all love what he has to share.
All right. Well, thanks, David. Thanks, Ian. This has been a great conference so far, and I really appreciate the mixture of science, acknowledging the challenges in bringing that science to our patient populations, and then also the crucial lived experience that we've heard from the past couple of days. So one of the items that we've heard repeatedly and is also certainly present in the literature is the importance of restoring hand function after paralysis to those who have lost it. So we've been thinking about how we can leverage new technologies and also repurpose some existing technologies towards that goal. So today I'm going to do a little bit of a two-part talk here. I'm going to share a little bit about our study with Ian using a brain-computer interface as a neuroprosthetic to improve both motor and sensory function. And then I'm going to pivot a little bit to show you how some of the things that we've learned from that trial have motivated us to to create some new technologies that we're working on today. So a little over 10 years ago, we had the idea to implant a Utah array in the motor cortex of an individual who had lost the use of his hands due to a spinal cord injury. The idea was that we could extract his brain activity when he was thinking about moving his hand in different ways, run it through a machine learning algorithm on a computer to determine exactly what he was attempting to do, and then stimulate his muscles using functional electrical stimulation to enable his paralyzed muscles to move again and evoke the desired movement. So when things worked well, he would think about moving his hand, and a split second later, his hand would move in the way that he intended. In order for us to do that, we needed a system that could move his hand in all sorts of different ways without having to reposition a bunch of electrodes like you would for a standard functional electrical stimulation system. And so one of the challenges in this project was actually building our own functional electrical stimulation system. You can see that FES system, the version 1, the original one on Ian's arm, in that picture. And so one of the key things there is having electrodes that cover the full span of the arm so that we can turn electrodes on and off and change the amplitudes all in software. And that allows us to control it then with our decoding algorithms. So this is a big collaboration. There were a lot of people involved. Batelle, where I work, and the Ohio State University Wexner Medical Center were the two mains. And our study participant, as I mentioned, was Ian. He was a C5 level complete injury. So his Utah array was implanted in 2014. He participated in that trial for over 7 years. At the time he was explanted, he was had the longest duration Utah array implant on record. I think he's since been surpassed, but he's still definitely in the top 2 or 3, I believe. So show you a couple videos. So our primary goal was to try to restore functional movement for Ian. So this is a good example of some of the activities of daily living that we would practice with him on a regular basis. So here you can see he's using the system to open his hand so he can get it around a bottle, and then he's going to get a power grasp. There it is. Pick up the bottle, pour the contents into the jar. He has to place the bottle back down without knocking it over, and then he's going to use a pinch grasp to try to pick up that little stir stick. It takes him a couple of tries. But he's going to pick up the stir stick and stir the contents. And so like I said, our focus at the beginning was very activities of daily living. And it wasn't perfect, but with practice, Ian was able to master a range of movements and execute them with pretty good speed. As we progressed, we were asking Ian more and more, what would he like to do with this system? And I'm going to show you this video. This one dates our study a little bit, but I think it's still pretty cool. So he said he wanted to be able to pull his credit card out of his wallet and swipe it through a reader so that he didn't have to rely on other people to make payments. So obviously that technology is a little bit dated now, but I think you get the point of trying to translate this not just to the standardized assessments, but trying to really focus on things that are meaningful to our patients. And I know we've talked about this before. There's been lots of videos recently of showing people playing games with their brain-computer interface, so we did some of that too. So this is Ian. He's running the Carla driving simulator, which is an autonomous car simulator. And so he's driving this car on a controlled course that we made for him. And he's in full control of going forward, going backward, and steering wheel left, right. So Ian has a complete injury. He has lost motor function in his hand. He also lost a lot of sensation in his hand. And so we did a set of experiments focusing more on the sensory. And so in these experiments, Ian was blindfolded, and we would touch him at various points on his hand and arm. So we touched forearm, we touched thumb, index, and middle finger. And so consistent with what you would expect with his level of injury, he was able to detect when you touch his forearm. He was able to detect when you touch his thumb. He has a zone of partial preservation there. He was occasionally able to detect touch on his index finger, and he was not able to detect any touch on his middle finger. But while we were doing this, we were also watching the neural signals through the Utah array. And what we found was for all of those touch events, we were getting very clear signals through the Utah array. And so we turned around our motor decoding and turned it into a sensory decoding algorithm, and we were able to very clearly separate out all those 4 different touches. So what do you do with that? So we decided that we were going to try to repurpose that signal and give it back to Ian in a way that he could perceive. So you can see the sort of image here. We took a little band, we strapped it around his upper arm where he does have sensation, and every time he got a touch sensation on his hand, we would decode that from the neural activity, and then we would use that to buzz up his upper arm, and it gave him a sensory substitution. And why is that important? Well, as soon as we did that and we started having him do functional tasks, we found, not surprisingly, that he was much better, much more accurate at those tasks, and was also faster at those tasks when he had that sense restored. So, as the trial progressed, we ran into what has been a common theme at this conference. So no one had quite done the things that Ian had done in the way that he was doing them, and so that made it hard for us to apply standardized outcome measures. So our collaborator, Dr. Bachbrader at OSU, came up with a useful framework for evaluating this type of neuroprosthetic device, and we call this the GAIN model. So I'm going to let this video play here. The G in GAIN stands for generalization to everyday objects. So we did a lot of stuff with these standardized objects from our outcome measures. So for instance, picking up a can, but we wanted our system to be able to pick up a full can, an empty can, a narrow can, a wide can. And so we would test with the standardized objects, and then we had a bunch of similar objects, not quite the same, to test for generalization. Second is the ability on standardized tests of upper limb motor function. So there are several tests. There's been a couple even since this slide was made. And they all cover slightly different things and do things in slightly different ways. The idea here was trying to map out all those tests and make sure that we are kind of covering the full range. The next one is independence and participation. This is more qualitative. This is getting at the quality of life, getting at measures of independence. And the last one is the neurological level of performance. And so this is one of the more exciting results from the trial, in my opinion. So after Ian had been using the system for a while, we reassessed his baseline function, and we found out that he was able to do things with his right hand, the one that he was controlling using the BCI FES system, that he wasn't able to do prior to starting the trial. Comparatively, his left hand, which did not receive any stimulation, remained at the exact same level. So you can see the performance improvements on task and prehension on the grasp. He improved on his 9-hole peg and pour tests. But more importantly, this was something that Ian reported to us that was translating into improvements in his daily life. Most notably, he was able to move into his own house and live a lot more independently than he was at the beginning of the trial. So this was really gratifying to us. I know it was gratifying to him. And it got us thinking to how we might be able to obtain similar kinds of effects, but without all the complexities and the cost of invasive surgical implants. And so we went back to the drawing board, and we're thinking through what might be a device that could do that for us. And we were really driven by these 3 principles that we think of as sort of the guiding principles for these closed-loop functional electrical stimulation therapies. So first and most important, we want to have continuous closed-loop control of FES for tightly coupled enhanced sensory feedback. So the system needs to be very responsive to the user's intent, not just to turn on the stimulation, but to maintain the stimulation for as long as the user wants, and then to be able to turn off the stimulation exactly when they want to turn it off. It's going to enhance their sense of agency or feeling that they're in control of the system. And this criteria really drove a lot of our technical performance criteria. We want the system to be used primarily for goal-directed rehabilitation. So we don't want to be doing the kind of FES that you might see where we're going to go back and forth 30 times, and the user might be dozing off as they're getting the stimulation. So they have to be actively engaged. They have to be, every time they're moving, they need to be volitionally attempting that movement. And that's to maximize the engagement of the reward circuitry and encourage neuroplasticity. And then we're also using a rehabilitation, a progressive difficulty in our rehabilitation. So we're constantly trying to challenge them. And as they master things, we up the complexity, so they're always having to push. Lastly, we want to make sure that the system enables an extensive range of functional movements for arm and hand. And so just like the first system you saw, our new system covers the entire forearm and electrodes. And that allows us to target the key musculature for complex motor control of arm and hand and leads to widespread engagement. So the system that we came up with, we call the Reclaim EMG FES sleeve. You can see it over here. It's a wearable system. It just has a single zipper that you zip up. It has 150 electrodes that cover the entire forearm. And these electrodes can do two things. So like before, they can stimulate, and we can, in software, change the stimulation parameters, turn different electrodes on and off, modify the amplitudes. But they can also sense using EMG. And one of the clever things that our engineers were able to figure out is how to do those at the same time. So we can be listening on EMG, detect a movement, stimulate with the FES, still listen to EMG in between our stimulation pulses, and that enables that continuous control so that we can tell when the user is not just intending to move, but when they intend to stop movement. This was from the bottom up designed to be easy to use. Our initial vision was using in a rehab setting, so the user would be able to wheel into the clinic, quickly don the system, quickly calibrate it, get an hour of rehab, and then go home. So you can see some data here. This is actually also Ian's data. So here he's doing a couple of different wrist moves, and you can see, I guess you can't see my mouse. There it is. So this is a single channel of EMG. You can see when the FES is on, we get these very large stimulation artifacts. And so we have some algorithmic approaches to help remove that. With the 150 channels, we get about 150 electrodes, we get about 75 channels of EMG data. And so you can see over in this panel, we get very distinctive patterns of activity for the different movements. And then the bottom panel here, you can see our neural network decoders processing that activity in real time, and the output of that probabilities are driving the stimulation. We can also visualize the EMG activity. So here's what it looks like for hand flexion, extension, and index extension. You can imagine this as the sleeve sort of laid out flat on a table. So the wrist is at the top, the elbow is at the bottom, flexors to the left, and extensors to the right. And then there's also a ton of information in that EMG. So in both stroke and spinal cord injury participants, we've been able to get dozens of motor units. We've been able to pull out muscle synergies, all of which have lots of interesting applications as biomarkers. So showing a couple examples in spinal cord injury for our EMG FES system. Here you can see 9 of the different kinds of functional movements that we've been able to get with some of our spinal cord injury participants and their different injury levels there. And then, so again, here you're going to see this is part of the grasp and release test. On the bottom right, you can see the EMG activity in real time, lighting up with the different movements. When you see red and blue circles, those are electrodes that are turning on and off for stimulation. Here for thumb abduction and key pinch. And this is a task where Ian, again, this is C5 complete, is attempting to pick up this paperweight. And you can see in the top, he's able to get his hand around it, but he's not able to generate enough force to pick it up and put it on that raised platform. But with the system, he is. This is another one of our study participants. He's a C6 incomplete injury, and here he's demonstrating picking up a mug, it's a weighted mug, and bringing it to his mouth as if he was about to take a drink. So we also do a lot of tasks where we're trying to simulate use at home. One of our ultimate goals is to build a system that people can take home and use it in their daily lives, doing things that they would be doing with their families. So here we're playing Uno with one of our participants. And you can see he's able to use the system to pick up the cards, and also, importantly, it stays off when it's not his turn. And so, like I said, we like the idea that you could be using this both as an assistive device to help you with some of these tasks, but then every time you're getting that closed loop stimulation, you're reinforcing that neural circuitry and hopefully getting some rehabilitation at the same time. So the last task here, again, this is another at-home task. There we go. This is a pill sorting task. So these aren't real pills. They're little colored foam blocks. But his task here is he has to sort these pills by color and by day. So you'll see he's picking up the task with a little key pinch. He also has the ability to do some thumb abduction to help his hand get in position. And then he's picking up those things and having to, based on color, sort them. And so you can imagine, this is a task that is very easy for us to modulate the difficulty up and down for him. We can make the pills smaller, we can add more colors, things like that. So we have an ongoing trial in spinal cord injury that's funded by the DoD, the CDMRP. And that's ongoing. I don't have a ton of results to share there, but I did want to share a few of our results in an early feasibility study that we did in stroke. So we had 2 stroke participants come in with chronic upper extremity hemiparesis. They did an 8-week protocol, 3 sessions a week, 1 hour per session. They were both several years post-stroke. And what we found was that they very quickly experienced improvements that they perceived to be beneficial during the 8-week therapy program, and that those gains persisted throughout the 10-week follow-up period. So again, these are gains, they were using the system in the protocol, but this is changes in ARET and upper extremity Fugl-Meyer without using the system. So these are rehabilitative gains that they had. And again, it's important for us to be collecting these quantitative outcome measures, but it's equally important to listen to these participants and hear the improvements that they say they had. So our one participant informed us that he was able to tie his shoes for the first time since his stroke after doing this protocol. And our other participant said she was cutting vegetables in her kitchen, looked down, and was using her right hand with the knife, which is something that, again, she hadn't done since her stroke. So last video to share with you. We can get it going here. So again, this is one of the participants. The first video you'll see, this is her prior to undergoing the therapy. This is part of one of her functional outcome measures. So it's a little ball bearing, and her goal is to try to pick up the ball bearing and place it in the tin that's above the box. And you can see we speed it up here. She has a minute to do the task, and she's not even really able to get started with her level. So then she went through the 8 weeks of therapy, and this is her after completing the therapy protocol. So I wish there was some sound on this, but you'll see. So I think we talked a little bit about holy shit moments yesterday. This was definitely one for us and our team. So I'll leave you with a few parting thoughts. First, solving these complex problems, it's okay. Solving these kinds of problems require really large multidisciplinary teams, and so I'd be remiss if I didn't acknowledge both our team and the several funding agencies who have helped enable this. I know several of you are here today, so thank you. I believe it was Matthew, I don't know if he's here, yesterday said, we should be thinking about combinatorial therapies to solve these difficult kinds of problems. And I think our technology is perhaps a good example of combining different sensing and stimulation technologies with closed loop algorithms. But I'd be very interested, and I'll be around the rest of the conference if people have ideas for how we might combine this kind of a therapy with some of the neuromodulation approaches that I know a lot of you are working on. And lastly, we do have some ongoing clinical trials with our technology in Columbus, Ohio. So if you or anyone you know is interested, feel free to reach out to me. My colleague Michael Darrow is here as well. You can reach us at the email address, or all our trials are also on clinicaltrials.gov and SCI Trials. Thanks.
All right, now amazing talk, and now you get to ask some questions. So we'll have Riki and Andrea and Dave come up here, and then Ian is going to grill you.
All right, so I think we got to see some great opportunities there, from using a device for communication, then using a device for lower extremity restoration of function, then upper extremity device function, and it really shows all of the impacts that BCI can have. Before we go to the audience for questions, I have a couple questions here for the panelists, both for Riki and Andrea. I wanted to know how you, both coming from the industry side, how can you balance creating a device that you can get out to the market, but also being able to innovate on that device for future generations and make it be something that isn't going to let the early adopters feel like they're left out by using a iPhone 1 when we're past that?
So actually, I'm pure academic. Don't come from the industry, so I guess I let answer, because what we develop, of course, we use different technologies, and we have this big partnership that we do with Onward that allow us to continue doing the clinical trials. But let's say, me personally, as a postdoc researcher, we aim more to see actually the outcome of allowing this technology for the participants.
So I'll mention maybe two things, and they've actually maybe, well, at least one of them was mentioned earlier today. So my training comes from, I worked for Tim Dennison, and so the model that he showed of developing a platform that allows you to continue doing research, continue doing early feasibility studies, but also be a product is kind of the model that we're adopting. And so you can do a lot these days and put enough memory, computational power in an implant to be able to do things with software unlocks. You can architect your software such that your software is actually sitting on an external device, and you kind of, maybe limit what's on the implanted device so that you can innovate a little more easily and without introducing at risk at all to the patient. So there are ways to architect your system and kind of go towards that software unlock that allow for upgrading or updating for someone that's been an early adopter. The other aspect in particular in BCI is I feel like I'm creating that interface, but I don't know that I think Synchron's strategy is to create the whole rest of the ecosystem. You saw I was already interfacing and interacting with iPhones. They can do that as a state of the art. I'm not going to compete with that. I know how to make implants, though, right? So can I offer what we can be good at strategically, and then an interface that can be standardized and used with the future VR headsets, the language learning models, the accessibility tools, and that ecosystem there. And I think you're on the BCI collaborative community, and some of that is about to make sure we're standardizing that interface so that then we can enable that ecosystem and that connection, and maybe also that partitioning of medical risk and kind of usability that maybe doesn't introduce risk in particular in this domain.
Awesome, thank you. And then I have one question for Dr. Friedenberg real quick, and then we'll open it up, so get your questions ready, please. Dave, I think it's really interesting, I got to experience firsthand both sides of the technology and got a lot of improvement out of it because I was able to participate in it for so long. How well do you think that will translate to other participants, whether they are using this in the clinic or have the use of this at home and are being able to access it as they wish?
Yeah, it's a great question. I think we've seen with a lot of the presentations that there's a lot of variability. Like, we know spinal cord injury is highly heterogeneous. So that's one of the things that we're trying right now with our CDMRP trial, is trying this with 12 spinal cord injury participants. We intentionally made our inclusion criteria very broad to try to help us define some of those boundaries of who might be a responder and who might not be a responder. But I think it's a really important question going forward.
Awesome. So do we have questions? There we go.
Hi.
5 minutes. Okay, great.
I have a question. I jumped right for it. So, Ian, I actually had a question for you. As an advocate and a pioneer, I'm sure you've experienced so much of which outcomes excite you the most, both that you got to see and that you talk about, and how can some of these studies, be enveloping of more PROs that are beyond word count or step count, that are individualized but can still do rigorous science?
Yeah, I think it's really important for me, and I mean, it was talked about during the lunch session too by John, some of those really little things that translate into a big thing at home. So one of the earlier tests that we were doing, we were picking up the bottle. It's full of a bunch of dice and pebbles because engineers don't like water around their electronics. I'm not really sure why. But just showing like a large grasp like that and then going right to a finer grasp with a pinch grasp, that can translate out to me being able to do, roughly like 60 to 70% of what I'd want to do with my hand. I don't really care if I can go and type fully independently on the keyboard or play the piano, but being able to do meaningful activities with my hands so that way I can have someone help get me up in the morning, and then I'm on my own completely until I need to go to bed again. And reducing that kind of overall caregiver burden and also increasing my independence is something that's huge. And that varies from person to person. Someone that might be just allowing them to stand to transfer themselves from their wheelchair to the bed. But for me, it was just those little things of being able to generate force out of my hands because I have arm movement from my shoulder. It was also really interesting, and I didn't expect this at all, of being able to get sensory information out of the motor cortex. And that's a big gap that needs to be filled in how we are able to control these devices, because if you want to be able to reach down and grab something out of a drawer without looking at it, or even if you can see it, if my hand was occluded by the object and I couldn't see if I was getting a good grip around the object, I wouldn't have enough confidence to know that I could pick that up and maintain that grasp, which is fine if you're picking up a block and moving it across in an occupational therapy task that you're doing in a lot of these clinical trials. But if you're picking up a knife and cutting vegetables, that's something that you don't want to drop.
So a question for Synchron. I actually have two. So for the stentrel, at some point, the wire of the electrodes needs to come out of the blood vessel, right? So there is an interface when the wire travels from inside to outside of the blood vessel. So do you have any real-world evidence to show the safety or adverse event related to that particular interface? So the second question is, obviously this technology is governed by where the blood vessel travels. Do you think there's a limitation in terms of what type of data you can record from the brain? Okay.
So can you repeat the second question? I don't know if I heard the whole.
Yeah, the second question is where the stentrel needs to go, governed by where the blood vessel travels. Like, do you think there's a limitation in terms of what data you can collect from the brain?
Gotcha. Okay, yeah, so the first question was about the interface and the exit from the blood vessel. So we are implanting in the venous system. Actually, most neurointerventional devices are implanted in the arterial system. But when you implant in the venous system, the blood flow is much slower, the pulsation is much less, and the adverse events and hazards are lower. So when you talk to a neurointerventionist and you're talking about a jugular puncture and going in right there, they don't blink. They're like, yeah, it'll be fine. So you hold pressure and create homeostasis, and and there's clotting there. And from an adverse event standpoint, we've just seen some bruising. So it really hasn't been a challenge in terms of that. And then let's see. I think your other question was about the location and the placement. So I think in creating this device and this system, I think there was a lot of understanding of okay, where can we place a device and what benefit can that give? And this happens to be a really good application for that. And the way that we're using it is we're translating, I can be thinking about tapping my toe or tapping my ankle, but I can translate that to an on and off, right, a binary. And so it kind of depends on what you're doing with that information and how you're translating that. If you're going to direct translate that and say, oh, well, then you have to do something with your ankle or something that doesn't quite work. But if you're saying, hey, that's a binary, that's an on-off, and therefore then I'm going to translate that and create that into some other action that's happening, then you have more possibilities. And so I think we've seen that, and even with the fairly simple lower electrode count, we're able to create pretty compelling user interactions, and then as I mentioned, we're interfacing with its ecosystem of accessibility and tools that are being made in the external sphere. And so that coupled together creates a compelling user experience.
All right, let's give everybody a round of applause. Thank you so much for being here.